that what's happened here is an escape from a lab, because we're dealing with a virus that turns up in the city, which has the lab that does work on SARS-like coronaviruses more than any other lab in the world. And that geographical coincidence has to be taken seriously, particularly when we find that the virus from the bat that they identified as being closely related to SARS-CoV-2 had been found effectively in their own freezer. And that's a starting point for a query. So by the middle of May 2020, the Chinese were announcing they didn't think it started in that market. Alina Chan was saying there's lots of evidence to suggest this thing is well adapted to human beings. And the geographical coincidence all got me interested in this being an open question, not a closed one, and one that needed further investigation. And the deeper I dug, the more emerged.
Okay, so let me summarize that. So the first smoking pistol, in some sense, as you point out, is the coincidence of the location of this lab, which studies exactly this kind of virus, and the outbreak itself. And that's a problem, right? So that means that it's reasonable to look at that and think that, well, it could have escaped from a lab there. That's the first conclusion. And that it has to be demonstrated in some sense that it didn't. And then, so that's a problem. And I can't see how that's anything but an incontrovertible problem. The mere fact that that lab is there and that it does research on those types of viruses and that that's where the outbreak was doesn't prove that it originated in the lab, but it certainly makes that a plausible hypothesis. But then you have this additional twist, which is, I think, more complicated for people to understand. And you detail this out, you provide some detail for this in the book, that this virus is somewhat remarkably well adapted to human beings. Now, there are literally trillions and trillions of different forms of viruses. And so obviously most of them aren't particularly well adapted to human beings, because otherwise we would have trillions of viruses producing pandemics through all the time. So it's generally the case that viruses are not well adapted to transmission in human beings, and that's true for the overwhelming majority of viruses. And so the fact of this human adaptation, or adaptation to human transmission, is something of signal importance. And so maybe you could walk me and everyone else listening through why a typical virus isn't adapted to human transmission and what it means that one is and how that develops.
Yes. The normal pattern when a virus first emerges into the human species is for it to be very difficult for the virus to spread human to human. It can infect someone, it can even possibly kill someone, but they're not very good at passing it on to people. The virus is not really very good at transmitting between members of this new host. Now, if enough time goes by with enough infections happening, then eventually it will get good at it. And that's what was starting to happen with SARS in 2003. It first infected people in the fall of 2002. By the spring of 2003, you were starting to see chains of transmission from person to person. And the reason for this is that the virus has to evolve. It has to change its genetic code in such a way that it can better fit the receptors on the cells of humans as opposed to the receptors on the cells of bats, or in the case of SARS, the intermediate host, which was a palm civet.
Will the lack of a red wave during the midterm...
So we should point out that the problem of transmission that a virus has to solve is no different from the problem of the virus determining in some sense or evolving so that it can live. Transmission in a virus is the same thing as propagation in the environment. And so this is a very knotty problem, K-N-O-T-T-Y, for a virus to solve. It's not simple at all because it has to not kill the host too quickly. And it has to be able to replicate within an individual, but then it not only does it have to manage those things, which is very difficult, it also has to figure out how to transmit itself with some degree of effectiveness. And thank God most viruses can't solve that problem. So this is a very thorny problem. And maybe you can outline for everyone how viruses do solve that problem and why most don't, and why the fact of human adaptation to humans is so important.
Yeah. So just to give you a good example, bird flu, which is a big problem at the moment in poultry flocks and wild birds, is not very good at infecting human beings. People can catch it. People have even died of it. If you're exposed to a huge dose by working in a poultry farm, you can get sick and you'll probably die, but you probably won't give it to anybody else. We've not seen any human-to-human transmission of that virus. This virus was good at transmitting between human beings from the word go, from the November or December of 2019. And the reason for that, we now know in molecular terms. It has a so-called receptor binding domain on its spike gene, which is well adapted to the human ACE2 receptor on our cells. Not very well adapted to other species receptors, which is intriguing.
So it's a specific human adaptation.
Well, not specific. It's not completely specific. It's quite good at lots of animals, but it's really good at us. Okay. And the other feature that it has that is very striking and very surprising is that it has something called a furin cleavage site. Now, this is a small section of the spike gene that is coded by about 12 letters of RNA genetic code, which has been inserted into it, compared with all its close relatives. And that gives it the ability to use a human enzyme called furin that is in all our many of our cells, in order to reconfigure the virus as it leaves the cell to make it, to as it were, prime it to attack another cell. So it's actually using one of our enzymes to spread in the body. This enables it to infect more tissues in the body and to multiply much more effectively. So it's the reason we're having a pandemic, is the furin cleavage site. If this virus didn't have that, it would probably have been pretty easy to control in the early months.
you said something very interesting, and this is another piece of the puzzle, I presume, is that compared to other viruses of its type, it has this interesting furin cleavage site. I've got that right. And you said that it had been inserted, and that's that's an anomalous statement, let's say. So there's something about this particular virus that sets it out against other viruses of its type, and it's this particular ability to use a human enzyme, and you described that as inserted. And so I presume that could be inserted as a consequence, hypothetically, of natural selection processes, or that there's other alternative explanations. So what do you see when you look at that?
What I mean by inserted here is that it's an extra piece of genetic information. We can look at 20 or 30 other very similar viruses, and we can line their genomes up and match them up against this one. And the furin cleavage site is not an altered bit; it's an added bit. It's an extra chunk that's been added in the middle of the spike gene.
So how do you differentiate between added and different? I presume that's a consequence of degree of divergence. So you can imagine that there's a set of viruses that are genetically and evolutionarily similar, so they stem from a similar source, and they have a predictable pattern of variation. And you're saying that this is an anomaly that exists outside that.
Well, you line them up. You align the genetic codes of the viruses, and in this particular part of the spike gene, they all align up quite well. The sequence is mostly the same. You can tell it's been added because you can align the genes of the spike genes of other viruses alongside each other and see that this is an extra piece of RNA, not a changed piece of RNA. It's about 12 letters long, and this spells out a sequence that allows it's called a furin cleavage site, and it allows the virus to use a human enzyme called furin to spread from cell to cell, from tissue to tissue, and effectively makes the virus much more dangerous and much more transmissible. It's the reason we're having a pandemic. If the virus didn't have this furin cleavage site, we'd probably have been able to get it under control very easily early in the pandemic. Now, what's interesting is that a number of Western virologists, when they first saw the sequence of this virus, said, whoa, it's got a furin cleavage site in. That's very unusual for a SARS-like virus. In fact, it's unique. We've never seen one with this before. There are other coronaviruses with furin cleavage sites, but not SARS-like coronaviruses. And they said, I'm afraid that suggests it might have been engineered. Now, they kept those thoughts to themselves. We only know about them now because of leaks of emails that have emerged more than a year later. But they got on a conference call at the beginning of February, US and UK and other virologists, about a dozen of them.
Who are these people, this they that you're referring to, and in the early stages of speculation?
Yeah, these are virologists, so senior virologists who've been studying this kind of virus or other similar outbreaks. There's about a dozen of them. But also on the call was Dr. Anthony Fauci, the head of the National Institute of Allergies and Infectious Diseases in the US and the main advisor to the president on this, and Dr. Jeremy Farrar, a senior advisor to the British government and the head of the Wellcome Trust in the UK, which had funded a lot of research of this kind. And they discussed on this call their doubts that this virus was natural and their worry that it might have been engineered. Within 2 days, however, that same group of virologists started drafting an article which was eventually published in Nature Medicine, saying it couldn't possibly have been engineered. The furin cleavage site will probably turn up in a wild bat virus. Well, it hasn't so far. And the reason they have given for changing their mind after these emails emerged showing what they were actually thinking in February is that the Chinese had announced they'd found a virus in a pangolin. You probably remember that. A scaly anteater that is trafficked because of the belief that it contributes to good medical health and so on if you eat its scales. It's not true. They're made of the same stuff as fingernails. You might as well eat your fingernails, but still, it's a widely held belief, and as a result, many of these scaly anteaters are trafficked into China. Well, it turns out that a university in China announced in February 2020 that they'd found a very similar virus, a 99% similar virus in pangolin. And people thought, right, case closed. We found the intermediate animal. We know what's happening. Well, there's 3 problems with that. One, when they eventually published the sequence of this pangolin virus, it was not very similar. It was 90% similar. That's not good enough. It's nothing like close enough. 2, it didn't have the furin cleavage site in. And 3, there were no pangolins on sale in Wuhan. So it couldn't have explained how the outbreak happened in Wuhan.
Those are problems.
Those are big problems. So we're in this strange situation where this particular feature has alarmed Western virologists, but they've kept the information to themselves. We didn't find out about all this for months, remember?
No, it's worse than that. It's worse than that because these virologists that you're talking about include Fauci. And so it's not just virologists. It's the virologists who end up being in charge of the entire response. And so the question that emerges for me there is that if they were concerned about this being a lab leak, then why the strenuous attempt to deflect? Now, there's 2 possible reasons. One is that they didn't want to move forward with their presumption that it was a lab leak without a smoking pistol, and that's fair enough because you might think, well, yeah, we're concerned and we're incurring, but we're not going to beat the drum about the lab leak till we're certain. And then there's whatever other reasons might be lurking in the background, and I suppose that's partly what we're trying to investigate. And so that would be the scandal that would emerge, perhaps, if it was a lab leak, and what that might do to Chinese-American relationships, and what it says about virology research in general, and God only knows what other host of explanations. And so, but it's very striking to me. So you've laid out a story that goes, well, first of all, there was a lab in Wuhan that was doing research that was strikingly similar on viruses that were strikingly similar to the virus that caused the pandemic. And that is the geographical locale of the origin of the pandemic. And then the virus itself has peculiarities that might indicate engineering. And so that's two pieces of evidence that are starting to converge pretty hard, and unlikely convergence. And then you have the virologists themselves, including those who are in charge of the response, or who will be eventually, also noting that this looks suspicious, to say the least. And then, for some reason, and in a great scientific journal, or at least a once great scientific journal, downplaying their own fears. And so, what's the motivation here? What's going on precisely?